Alliance. And I would like to give you a little bit of a background on how we made our connection with this project. It actually goes back to about 2016 or so. The Quassia Creek Watershed Alliance was partnering with Hudson Valley Regional Office on a grant to develop a citizen science model to sample the Quassia Creek for microplastics contamination. The Quassia Creek, for those out-of-staters, is one of the tributaries to the Hudson River flowing through Newburgh, New York. Our executive director, John Gebhardt, he also saw in the Hudson River Almanac a mention of microplastic project that was being done by the Hudson River Park Trust. With that, we got 2 contacts. One of those contacts provided us with a 3rd contact. When I spoke with this 3rd contact, I was told about their project, which involved the trawling of the lower Hudson River in the harbor, an area around Staten Island and Sandy Hook, New Jersey. This was not exactly what I was hoping for, but because what we couldn't do is, how do you do a tributary? And when I spoke about our grant proposal to these people, and asked them about the technique for sampling tributaries, it turned out that one of the original 2 contacts was a Marist College student who had developed a microplastics project that included a collection technique in this analysis of the stream samples. So John and I met Ian at the Duchess Diner, and Ian was graduating and heading off to grad school. So he gave us a name of another Marist College student. Jason came to a meeting that we held at the Algonquin Park, which the Quassia Creek flows right through there. And he showed us the technique for stream sampling. Later, we were given a tour of the Marist lab, and Jason sampled the Quassia Creek in 6 different spots and provided us with a baseline data regarding the microplastic contamination of the Quassia Creek. With Jason was another Marist College student by the name of Carter. Carter and Jason came to one of our meetings at Mount St. Mary College in Newburgh to present their findings. Well, then Jason graduated and Carter was then the contact. So in the summer of 2019, we met up with Carter and Zion, who is his advisor, and Gabi, who is our presenter for tonight. And we met at the Chadwick Lake. And now Gabi is getting ready to move the microplastic project forward. Carter, who was to provide us with his final presentation in the spring of 2020, when that did not happen, and he graduated, and then I was able to remain in touch with Zion. And after a few emails, this webinar came about. Contacted Emily, and everybody was on board. So that's pretty much the little background, and it's awfully nice for a community group like the Quassia Creek to have the Hudson River Watershed Alliance, the larger group that Emily heads up, plus Marist College, coming together like this for this opportunity. I hope it serves as a model for future events like this, and we get to understand the water issues that are in the Hudson Valley and elsewhere, obviously. And thank you all. Gabi, it's all yours.
Hi all, I'm Gabi. I'm just going to share my screen. So, hi, my name is Gabi DeGennaro, and along with my advisor, Dr. Zion Klos, we did this research project called Identifying Microplastic Accumulation Hotspots and Sediment Associated with Stream Outflows into Lakes and Estuaries. So as I go through the presentation, I'm going to explain a little bit more of what that title means. So first, who we are. As I said before, my name is Gabriella DeGennaro. I'm currently a senior undergraduate at Marist College. I'm studying environmental science. I began this research project in July 2019 under the advising of Dr. Zion Klaus, and I was working with Carter Hsu. In addition to this presentation, I presented my project at the American Geophysical Union Fall Meeting 2020. I plan on creating a manuscript with Dr. Zion Klaus in the future, and I look forward to continuing graduate level research in the future. I also want to give a special thank you to my peers in my hydrology lab group. Some of them are here today. They have helped me with fieldwork and lab work, specifically Carter Hsu, Katie Samaro, Olivia Fabrizi, and we all help and support each other in our own little scientific endeavors, and it is run and operated by Dr. Zion Klaus, so I'm very thankful for them. So what are microplastics? According to NOAA, microplastics are defined as any type of fragmented plastic that measures less than 5 millimeters. And they also are the most common type of marine debris and found very heavily in our waterways. So the debris can either come from a primary source or a secondary source. Primary sources are cosmetic products, and secondary sources are usually from the breakdown of larger sources such as water bottles and bags. And there are also 5 subtypes of microplastics, which are fibers, foams, beads, fragments, and nurdles, or also known as pellets. So it's not my picture, but I included a picture down below so you can just quantify them and see how they look. So these aren't rare. Microplasts have been found in both small and large aquatic animals, from plankton to whales. They're in our seafood, and they're even in our drinking water. So to start with our objectives, we had to better understand the movement and accumulation of microplastics in aquatic environments as it relates to water flow velocity. So we wanted to see how the water speed in a certain point in the estuary or tributary attributed to the amount of microplastics in the sediment. And then we wanted to map microplastic and sediment at the outflow creeks to ideally find spatial hotspots of accumulations. And we looked specifically at Chadwick Lake, Wappinger Creek, Wallkill River, Rondout Creek, and Esopus Creek. So that was just to hopefully find a pattern. So next we have our sites, and the first site was Chadwick Lake in Newburgh, New York. Chadwick Lake is a reservoir supplying water to the town of Newburgh in Orange County. This was our site map. It connects to the Quassia Creek, and we took measurements at each red dot along the lake. Next, we took sediment samples from Esopus Creek in Ulster County, New York. A sample was taken at the mouth of the tributary, the midstream, and upstream, all before the intertidal slackwater zone. And again, we implemented the same sort of figure here. So this is Rondout Creek, and we also took sediment samples at the mouth, midstream, and upstream. And same for Wappingers. So we took, again, sediment samples at mouth, midstream, upstream, all before the intertidal slackwater zone. So the methods that we used for sampling Chadwick Lake and the 3 creeks. For Chadwick Lake, we started from the mouth of the creek, which was site A on the map, and we collected sediment from the bottom of shallow water using a shovel. So obviously the shallow water didn't last. Then we had a paddle boat. So we paddled out, we had measuring tape, and every 50 meters we designated a new site. We dived down and we scooped surface sediment into a 100 cc tube. And then for Esopus, Rondout, and Wappingers Creek, starting from the mouth of the creek, sediment was collected via shovel because it was all pretty shallow, and it was collected approximately every 300 meters. So these are some photos from the field and lab. As you can see from the left picture, we went in the middle of the July of 2019, so a lot of water chestnuts, and I am with the snorkel on the left side of the boat, and my friend Carter, who was helping me, is in a wetsuit because we got stuck in the paddle boat, so he had to get out and push us. So that was fun. And then to the right are just two pictures of me in the lab. So next we have the lab analysis. First, we took each individual sediment sample, homogenized it, mixed it, and then sieved it out. So we used two different sieve lengths. For Chadwick Lake, we did 2.35 millimeters, and for the three creeks, they all went through a 4.75 millimeter sieve. The sediment that passed through their respective sieve was then weighed out to 10 grams. It was also dried. So after that, a density separation or an SMI test and a wet peroxide reaction was performed on each sample. And the wet peroxide reaction left only the microplastics in the beaker, which were weighed and then plotted on a graph, giving us our results. So, of course, COVID affected us. When the labs were open, originally we were planning on dyeing every microplastic sample with Nile red, and with the Nile red being soaked into the microplastics, we could see each individual piece under a fluorescent microscope and have the ability to count them and decide which subcategory of the 5 types of microplastics they can fall into. Fortunately, due to the unexpected close of campus in March and limited lab restrictions throughout the fall semester, we could not continue this process. So first are the results from Chadwick Lake. So down on the x-axis, we have the site, which is, we start from A, the mouth, and I'm just gonna go to the site map to orient you all. So we started from A, and then every 50 meters we moved. So it was 50 meters, 100 meters, 150 meters for D, 200 meters for E, 250 for F, and then 300 meters for G. So that's reflected down on the x-axis, and then on the y-axis, we had microplastic concentration in sediment, proportion dry weight. So this was the proportion of microplastics per the original 10 grams of dried sediment that we weighed out. So as you can see, it starts out very low at 0.00034. It slowly creeps upward and then it reaches a peak at site D, and then it falls back down to sort of a median value. And as we can see here, site D falls right at the end of the water chestnut patch and starting to get into the Eurasian milfoil patch. Then we have the Esopus Creek. So the Esopus Creek, we started out at the mouth, had a low concentration, and then it peaked in the middle, midstream, and then it fell back down during the upstream. So you can see on the map, we started out with the mouth, it peaked right here in the midstream, and then it fell before the intertidal slackwater zone. We have the same results for Rondout too. We found that similar pattern of it peaking in the middle, starting low, ending low, and that definitive peak right at midstream. And same for Wappingers too. Started low at the mouth, definitive peak at midstream, and then fell back to median volume around upstream. And it's all before the intertidal slackwater zone. So our takeaways from this are, across all 4 systems studied, we did find these spatial hotspots of relatively higher microplastic concentration in sediment. So they do exist. All 4 peaks in the microplastics concentration were located slightly downstream location of the transitions from higher gradient stream flow to lower gradient stream flow of lakes or intertidal water. So kind of that median value as I was talking again. And these findings suggest that reductions in velocity and energy affect microplastics for their unique densities and shapes in novel ways that are similar but not spatially identical to accumulation patterns of other natural minerals and organic sediments. So Zion's going to take over from here.
Awesome. Thank you, Gabi. So as you can all see, Gabi's done some great work here, continuing on with the work of the lab group, and has really taken it to a strong level in terms of presenting at an international conference with this work recently. So again, big kudos to Gabi for all the great work she's done there. I just wanted to put together a little conceptual figure here for the audience to kind of get a sense of some of the processes that we think are controlling this pattern that we've seen at all 4 sites, where we see this definitive peak in the concentration of microplastics in the sediment. So I think it has a lot to do with this transition from a flowing river environment or like a fluvial environment in the stream or creek, and where you have a suspended sediment coming down as a source of that sediment from upstream areas. Then you hit that kind of rapid decrease in water velocity, which, you know, in those maps was symbolized by that purple triangle, which is everything downstream of that is the intertidal slackwater zone, where we see water flow both directions. So essentially the last mile of each of those rivers before they empty into the Hudson, or in the case of Chadwick Lake, where we go from Quasaic Creek, where it empties into the lake, and then you hit the lake body itself. And it's not surprising in anywhere in the world that you go from a fluvial environment into a low velocity water environment, you're going to get deposit of sediments. That's how deltas form. And that sediment is going to be primarily dominated by mineral sediments, but those are quite usually higher density than what some of our plastic sediments are coming in at, particularly the plastic sediments that we studied in this study, which are the ones that are less dense than the mineral sediment, because they were floated up in our density separation technique in the lab. And so as you move from this higher energy environment to a low energy environment, you're going to have a certain amount of time it takes for the sediment to fall out of suspension. And for your higher density mineral sediments there on the bottom, you're going to have a peak location where the majority of those are falling out. And that's going to be controlled by the density of those, which will vary, you know, for mineral sediments, it's usually between 2.7 and 3.3 grams per cubic centimeter. And it's going to also vary based on the shape of the sediment, with clays being more flat and some of your larger sediments being more spherical. But in general, you're going to see a peak, and that's going to drop off on either side. And that's how, you know, sedimentation is going to work anywhere in the world where you switch from a high to low energy environment. Now with the plastics, though, we're going to be looking at densities that are lower than these mineral sediments. And so we're looking at anything between 1 gram per centimeter squared, because anything lower than 1 would just be floating or in suspension in the water, and anything up to what we're seeing with those mineral sediments, because anything that is more dense, and there are plastics that are more dense, we're not going to be able to bring those out of the samples using the methods we have in the lab, which require a variation in density for the plastics to separate out. So we're really looking at all the plastics between 1 and 2.7 grams per centimeter cubed. And those, because they're a lower density, are going to stay in suspension for longer. And so that's why we think we're seeing this peak, is if you look at the above part of the graph, we have this kind of 3 locations that correspond to what Gabi and Carter's data we're looking at, where we have our highest proportion of plastics in sediment at that intermediate location, even though there's necessarily not more sediment overall being deposited there, just the proportion of plastics is higher. And it's going to be lower as you go farther towards the higher energy water upstream, and lower as you go farther downstream, as many of this sediment has already fallen out of suspension. And so that's why we think we're seeing these kind of concentrations of plastic hotspots at the locations where they're occurring. And that's different than where the sediment overall concentrates. And so that's why you get these higher concentrations of the plastics themselves. And I think, as from a management perspective moving forward, I think this is really useful to say, if this is a process that we see in these 4 different examples, and this is something that might be happening worldwide in terms of plastic deposition, then we have a better understanding of, hey, there's actually a way that plastics are already naturally being separated out from the other sediments in the environment. And we can get a little more bang for our buck if we decide, hey, we want to go in and remediate plastics in the sediment. We can find locations based on where these transitions from higher energy to lower energy flow velocities are occurring, and go in there and preferentially remediate those sediments, going in and dredging them up, and deciding what that might be in the future if we get to a place in a future world where there's a lot more priority towards cleaning up these plastics in the environment. So big picture, what we really want to show with Gabi's research is that there might be a pattern at a global scale where there is plastics sediment already preferentially being sorted out in this way. And so that's what we really want to show with our kind of introductory research here, and show that we easily see this in 4 examples here in the Hudson Valley, and only 4 examples that we've looked at. And as far as we can tell, the only 4 examples from other people. We haven't seen anyone else that's done a study like this, looking at these spatial changes in relation to higher to lower energy water. So thank you all for giving us a chance to share this with you. I just want to go to the next slide here and just let you know that, Gabi, yeah, great. As we move ahead, we also want to think about plastics and in the relation to vegetation. Both, I have students doing work in the main stem of the Hudson, looking at water chestnut and some aspects of that. And also in Chadwick Lake, we saw a transition from water chestnut to Eurasian milfoil, also corresponded to this highest peak. So there might be overlaps in terms of vegetation controlling microplastics, though those patterns didn't show up for the 3 estuary systems. Or there might also be a situation where certain vegetation is able to grow for the same physical reasons that control that. Those physical reasons also correspond to where this plastic is falling out of suspension. So there's a lot to still be understood there in terms of relationships with vegetation, but we think overall there's a strong relationship with just the physical system and its potential control on these lower density plastics. And we also were curious about if you looked at this system in places where we have particularly like point sources of plastic pollution, say like a wastewater treatment plant, versus more non-point sources where you have like distributed septic systems or distributed inputs from any potential trash dumping or just trash that's making its way from litter on the roads down into creeks. And with that, I think we're gonna call it good for now, and that gives us half an hour still to go through a little Q&A session where Gabi and myself can answer questions for folks, and I can hopefully lead into some more discussions. And I believe that Emily is going to head that off for us and kind of help with that process.
Emily, I'm just going to stop sharing my screen.
Good. Great. Well, thank you so much, Gabi and Zion, for sharing that information. Just to head off one potential question, we are recording this and we will be posting this presentation. So if you have other folks who you think might be interested in this topic, we'll be sharing that on our YouTube channel. So if you have any questions, please go ahead and put them in the Q&A. We've got a bunch here already. So question about your methods. How were the sieve sizes decided?
So, okay, I'm off. So we only had so many sieves in the lab, but we wanted to get 2 different values that are both under 5 millimeters. So we just chose the first 2 that fell under. So that was 4.75 and 2.35.
Great. Were you able to remain in an open channel for all of the sampling, specifically for the tributaries to the Hudson River, I think?
Yeah.
Yeah. Do you want to take this one?
Yeah, so Gabi wasn't involved in sampling there, but I went out for the Wappinger Creek site with Carter and another student, Ellie, who helped him with that. And yeah, so he was sampling in anything that was essentially deep enough for water chestnut to be growing and right on the edge of the active channel. So the criteria was we want to be near the mouth where it hits the Hudson main stem. We want to be up at the last point of the intertidal zone, usually about a mile up the creek, and then somewhere halfway in between. And we want to be as close to the middle as we can, but still know that we can get down to the bottom. And we use, for most of those environments, there's water chestnut growing on the edge up to a certain depth where it gets too deep or the currents get too high. for the water chestnut. So we used that as a kind of guide to decide, okay, we're going to go right to the edge of where that is. So the deepest location, and then he would dive down and collect a sample the same way that Gabi did.
Great. When you were doing that, did you or your student try to measure stream velocity, or are there plans to look into this further, thinking about the correlation of the velocity with the location of deposition?
Yeah, so so our main goal was to look at the relationship between this transition from a high to a low velocity environment. But actual velocities are pretty tricky to get at in these estuary systems in terms of just the number of sample times that would be required to go out there at different tide periods in terms of tide on its inflow or outflow. Definitely something that's possible, but I think we don't necessarily need it to tell the story, because we can say this is where we essentially go down to the elevation of the Hudson River, and we hit bi-directional flows in these estuary systems. And the same with the lake. It's where we started right where you have a stream coming in, where you see a elevation change at the surface to a constant elevation at the lake. So yeah, with additional data, I think that could help elaborate further on the story, but I think we have the big piece of it figured out just with using that sampling criteria of flowing water versus intertidal or slack water.
Great, thank you. How did the spikes in microplastic amount at each location compare to the population density with each transect? And I guess I have a similar question that I'll just tag onto this in asking about how each of those different water bodies compared to each other. I don't know if I got a note that said my internet was unstable, so hopefully you heard what should I repeat it?
Yes.
Yeah.
You heard it? Repeat it?
Yeah, we heard.
Oh, okay, great.
All good.
Perfect.
Yeah. So how they differed was Chadwick Lake. So I wasn't there for the creeks. I wasn't there to sample, but I was there to sample for Chadwick Lake, so I can speak on that. Chadwick Lake, you could tell it had some flowing water right at the mouth of the Quasaya Creek, but immediately after it was still water. It was very open. Then we reached the water chestnut beds and the Eurasian milfoil, which were not in the creeks. So that's what I can speak on that. I don't know if Zion, you want to speak on the creek?
Yeah, the population density question, I think, is important for understanding the overall concentrations of the plastics potentially. But I think that would really be dictated by the locations of wastewater treatment plants in particular. The Rondout Wallkill watershed is a massive watershed that's going in there. Esopus is not small either, and Wappingers also is quite large. So like Quasaya is the smallest, but in each of those environments, I think it's a good example to think about. Quasaya does have a very small estuary system in the last maybe 50 meters of Quasaya Creek where it hits the Hudson. But in upstream locations within Rondout and the Wallkill, in Esopus, definitely throughout Wappingers, there are places where you have other impoundments of water and you hit slack water areas. So likely these little hotspots occur throughout the watershed wherever you move into some of these impounded water bodies, where you go from a flowing stream into a still lake or reservoir. Related to population density, that would be a whole separate study that could be done, but I think you'd really have to consider how many different impoundments and where they're located throughout those watersheds, because those are each going to be accumulating sediments throughout them as well. You think just like Wappingers Falls and the lake there above the dam, that thing is almost entirely filled with sediment now. It's hard to get a canoe through there much of the time. So you can imagine as these different impoundments have filled up over the last 200 years of being in existence from when we've built a lot of these dams, that there's going to be little hotspots. And we can hopefully through this understand that there is going to be a potential location where the density of plastics is the highest, as opposed to being evenly distributed throughout all the sediment. And it's because of this difference in density we think this is happening, which makes sense. I don't think anyone would have thought that wasn't happening, but we're actually able to show that, and show it across sites that are both lakes and estuaries. But the actual values in relation to population and use, we need a lot more data to start making sense of the kind of inputs from the human side and what that means. And a lot of people are working on that. That's definitely happening. And not just the plastics that are dense enough to be sediment, but the plastics that are lighter than water too, that are flowing through these systems that we've had students work on in my lab group and are working on in a lot of other places too.
Great, thank you. Yeah, I noticed on some of the graphs, it looked like the Esopus values were almost an order of magnitude, or maybe 2 orders of magnitude higher than the Chadwick Lake. You know, that's obviously, Chadwick has a much smaller watershed than the Esopus Creek.
Mm-hmm.
I can see that. Okay, great.
Yeah, looking at population density, I feel like would help understand that a lot. We didn't.
So I think you already answered this question about what would you predict for hotspots if you continued to sample further upstream, right? We'd expect them throughout the watershed system in places where the velocity of water slows down. Okay, good, making sure I understand that too. How are microplastics remediated? So is there a method for dredging that citizen scientists could help with? Is this location complicated by larger pieces of plastic that are caught up in more dense sediments?
Yeah, that is a good question that I personally am not that familiar with. So I can't speak too much to that. I have not heard of many plastics projects on the sediment side of things. I've heard people doing work in stream for suspended plastics and other water bodies. And especially in the ocean, there's some really awesome remediation projects for out in the ocean for suspended plastics. Sediment in general is a lot more expensive to remediate than in-stream water or anything suspended in water. But I think if we can show that there's certain locations where it's worth it, that we might have more interest from communities and municipalities and state governments potentially in the future to clean these up, especially as we start to show more of the environmental and human health impacts that some of these sources can, and the issues they can cause. So it's really only been something we've even had accumulate over the last 50, 60 years in a big way. And we're just starting now to figure out where it is and what are the big issues that it's causing. So, yeah.
I still feel like the remediation side, especially for microplastics, are new. And even macroplastics, I feel like it's only maybe within like the last 10, 15 years that people are starting to do anything about it. So it's definitely a future issue, but I personally haven't heard of any microplastic remediation projects.
Great.
Yeah, especially on the sediment side, I have not heard of those things.
Can you provide examples of plastics that are denser than water that would be collected in these sediments? So distinguish between those lighter microplastics that are in suspension versus the ones that are falling out into the sediment?
Yeah. Do you want to go for Gabi or?
I was just gonna say it's probably related to type of microplastics, like the 5 subdivisions that I was saying before. So fibers are probably going to be less dense than water, but something like pellets or the fragments, those would have a higher density. But Zion, if you want to take it further.
Yeah, I mean, there's a whole plethora of different plastic types in terms of their chemistry, but one of the most common ones would be like high density polyethylene, HDPE plastics. Those are really common for a lot of the pellets that we see showing up there, because it's a really industrial plastic used for making containers and barrels and used for a whole variety of some of the handheld things we think of in terms of plastics. And we have found fibers as well in some of your samples, right, Gabi? Like from what you could see under the microscope. And so, yeah, it's not just, yeah, I don't think there's like one particular industrial or human source that's particularly heavily related to the higher density plastics that are showing up in sediments. I think they're spread around a lot of different uses, and there's lower density and higher densities being used for a whole variety of things out there.
Great. How many samples were taken for each of those data points that you showed?
So for Chadwick Lake, at least, we collected 10, well, we collected 100 cc tube, but it was 10 grams of sediment that we weighed out. So I did it for 2 different times on each site. So there was 2 microplastic samples for A, B, C, D, and E, and F, and G. And I'm not sure with the creeks, but from what I counted and from what I did, I only did 1 for each site. So mouth, midstream, and upstream.
Yeah, I think when we present it in the paper, we're going to make a combined figure. So it's essentially the 3 different creeks all in one figure, showing that, you know, the mouth is low, middle is high, near the upstream point where you finish the slack water is low again. So yeah, so they're essentially a N of 3 for estuaries with each data point, and N of 2 for the lake, except the lake has a higher resolution of information, you know, being more spread out across 7 sampling locations.
Great. I really like this question. Were there any surprises in your work? Did your findings confirm your hypothesis? And then the second part of that is, do we know if there's a correlation between plastic deposition and the health of the water body?
So I was surprised because at first when I was going into this project, I thought that it would be more in a still water because that's when they have time to float down and deposit. But I didn't expect them to be at that strict transition zone. I expected it to maybe be like a linear, just straight line. So that was very interesting to see. And in terms of health, I'm not sure exactly. Carter was actually taking samples of catfish and dissecting their stomachs to see how many microplastics he can find in there. So that can kind of relate more to health. But in terms of sediment, I'm just kind of seeing what's there and what's not.
Yeah, Carter's original goal was to relate these estuary systems, look at the 3 in comparison in terms of accumulation within catfish as well. And unfortunately with COVID, we hit a really big hurdle in terms of finishing up the lab work. He'd done all these digestions, but hadn't got to the last stages of what we needed for the essentially the fish gut purification down to the plastics. And so so we kind of stepped back from that and just focused on the sediment samples that were collected. Since in both Chadwick Lake and in the estuaries, we had sediment samples and they told a similar story, one at higher resolution, one at lower, and one in intertidal and one in a lake. So we decided to focus on that for Gabi's story that she's trying to tell. And we still have those other samples. So down the road, we have the potential to look at those in more detail if we'd like to, if we have a student that wants to expand that further. And a lot of the research questions depend on finding the right student that's interested in doing that as their project. Since, you know, as a hydrology lab manager for all these great undergrads doing projects, microplastics is one of, 7 or 8 different projects I have going related to water bodies in the Hudson River Valley area. So,
Great. Okay, I'm going to combine these 2 because they really work well together. So the first, I think, comment question is intriguing that in Chadwick, the peak concentration was just before the milfoil bed. I speculate that this submerged macrophyte acted to slow water flow, thereby enhancing deposition of the plastics. Okay, number 2, different person. Can you speculate on the role of macrophyte density in controlling the location of microplastic deposition hotspots?
I definitely think that the macrophyte population increased to slow down the water to increase the microplastic hotspots, because I wrote it in my future thoughts for future work that I was reading more, and also that there's some type of fertilizer that includes plastic pieces and it helps plants grow. So I wonder if it was more of like a symbiotic kind of relationship when it deposited into the sediment. But I definitely think that the macrophytes slowed down the water flow, which helped the microplastics stop and fall down into the sediment.
Yeah, and yeah, I think the relation with the macrophytes it's much more complex than what we have a data set to talk towards. But yeah, I think it's interesting that the peak in Chadwick at least is right at that transition from a really water chestnut dominated zone in the lake. You know, it was so thick that
Yeah,
we
bad.
had the rowboat thanks to Jack's help, and we were in there, and at one point it was like me on one oar and Carter on the other, and Gabi trying to keep us straight on like the Transect line, and us just not moving, to the point where Carter gets out. He's in a wetsuit, and he's in like 3 feet of water, just surrounded by the thickest water chestnuts you can imagine, and just like every shove as hard as he can push. And he's on the crew team here at Marist. He's a strong guy, and every shove moves the boat about a foot, maybe 16 inches, and we make our way the whole, you know, what is 150 meters through the water chestnut bed, doing that. So yeah, so that's at the surface, but underneath that water chestnut, compared to the milfoil, I could see that, yeah, there might actually be abilities for water to flow faster underneath it. And then once it hits the milfoil, there's a lot more vegetation dense deeper in the water column, that could slow things down, and we maybe see a corresponding spike there. Now, that might be happening in Chadwick, but this hotspot concentration we also saw in the 3 estuaries, and in those 3 estuaries we didn't have any transitions in vegetation like that, and all those vegetation was controlled for, because we were essentially sampling right on the edge of the main channel where the water chestnuts start for all those spots, both near the mouth and up farther upstream, until we get out of the intertidal zone. So yeah, there could definitely be interactions with the vegetation in those environments, but the way that we sampled, we would have controlled for that, and we still saw those spikes. So I think overall findings still hold up, that it's not vegetation alone that's doing this. It's more the physical environment's probably dominating, but vegetation could be relating. And it could also just be that vegetation is transitioning in as you go from higher energy to lower energy and deeper water. And those transitions happen to meet up with where some of our plastic transitions are too. Not necessarily they're both controlling each other, but they just both are codependent on the same physical system. So.
What is known about microplastics in the Hudson River sediment? How might that compare to the tributary sediment?
So I'm not really sure about the Hudson River exactly, because I didn't sample the Hudson River, but I know that I've seen many pieces of macroplastics in there. So as it flows into these tributaries and estuaries, I can assume that it stays there and breaks down a little bit more. So I don't know if you have any information about microplastics in the Hudson River itself.
Yeah, I would speculate. I haven't studied it myself, and I don't know of a study for sediment in the Hudson River. I know of suspended work doing for suspended plastics, but for sediment, my understanding is that the sediment and the bottom of the Hudson, and I've seen it with like we have a radar system where we can look at the actual sediment. You can see like, some of the shipwrecks down there, and you can see these big dunes of sandier sediment moving along the bottom. I'd imagine that those sediments are going to have concentrations of microplastics at similar levels to what we're seeing coming out of these estuary systems. But I don't quite see the same process occurring in a physical sense that would concentrate them necessarily in the same way. I think they might be more evenly mixed throughout all the sediment down there, at least the top few inches of sediment, which is the really mobile sediment. And a lot of it below that kind of stays put. And then there's stuff above that as the tides move back and forth and those currents pick up, that sediment moves back and forth. So I think in other estuaries throughout the Hudson, we'd probably see similar patterns of concentration points. But then once you get into the main stem, my guess, and this is just a guess, is that it would be more well mixed throughout the system because that whole main stem is experiencing the same huge shifts in velocity throughout the day as those tide shifts back and forth. Though there are some embayments and things that aren't necessarily associated with estuaries where we see a lot of water chestnut beds on the edge of the Hudson. And those might also be places where we go from higher energy to lower energy water. You might see concentrations underneath some of those water chestnut beds as we have a lot of those, so we've especially have a lot of embayments on the edge of corners where the railroad has come in and kind of cut the corner off a little bit. And you'll have places where water can flow in to an embayment on the other side of the railroad track between the railroad track and shore. And then there's like a culvert or two that gets you underneath there. Taking some canoes underneath those are fun because you can get trapped on the other side if the tide goes up. But that would be a place where I would expect you might see a big concentration of microplastics. We also see the same, just increased sedimentation in those locations. And so I could guess that if you go a certain distance from the main stem where you kind of come in from the underneath the railroad track culvert, there might be like a half circle of higher concentration microplastics if the same processes that we're seeing at these locations hold up, across the system. So that could be something exciting for future investigation too.
Were there any increases of microplastics captured during a specific season or time of the year during the study? Any correlation between wet events or after wet events?
Well, we took all of our sediment samples in the summer months, so we didn't sample it any other month. But if I were to speculate, it would more affect plastics in suspension, I feel like, the location, because if they're already deposited down into the sediment, I don't really see a rain event really moving them out. It could if it's shallow water, but in terms of the bottom of Chadwick Lake, I don't see it really affecting that.
Yeah, and I think the sediment deposition is going to be really episodic. That's just what we see in geology in general. You have a big storm event, like that big rain on snow flood that we had back around Christmas time here. I probably deposited a lot of sediment out into these estuaries, and there's probably a layer that corresponds to that big flood event and past flood events. But I think the process that's separating out the microplastics would be similar across all those and show up in a similar way, though the intensity of that event might actually shift where that peak in plastics is. So, that I think that's a good point. We could just be seeing a peak corresponding to the most recent major event to deposit sediment, and that peak might actually not be the same location. You go to the next layer down because that storm might have been a lot bigger. So flows were stronger. They pushed plastics farther out into Chadwick Lake, for example. And so that might be farther down. So that would definitely be something I think probably I'd say my most important way to think about the implications of this particular study is if you did a stratified look and went out and cored in those lakes and looked at, say, if you go down every 5 centimeters, every 10 centimeters and see, does those patterns hold up or do those patterns shift around as different size storm events have deposited different plastics? So that would be like the study number 2, I would say, off of the first one, just showing that these hotspots exist.
Great, thank you. And so Newburgh gets its drinking water, or the town of Newburgh gets drinking water from Chadwick Lake, right? the question is, where is Newburgh's water intake relative to Site D? And I'm sensitive to, source water protection, and maybe you may or may not be able to disclose the actual location of the water intake, but wondering if maybe you could speak to this as a drinking water source and what implications might be for that.
Yeah, maybe let Jack take the first go there, since he's the expert on the watershed.
Not exactly. That question I really don't have a good answer for at all. There are some people in the audience here that I can see names that would be a lot more familiar to that than I am. So if anyone from there could help us out with that answer, I appreciate it.
So if anyone out there is in the audience and feels like they can help answer this, please write into the chat and make sure it says panelists and attendees, and help us out with that.
But just to clarify, the data we were looking at was sediment at the bottom of the lake, and not the water that not plastic suspended in the water, which would be what was being intake for the drinking water system. So. Yeah,
just to add to that, Zion, when we began this with the Esopus Creek and Marist College, it was the suspended plastics that we were looking at, and what we saw Ian and Jason and even Carter got involved in. Gabi's sediment sampling was entirely different from the other ones that we had gotten first involved with.
Yeah, and I think that's actually a great explanation for why we kind of got to this project, because it had already been a pretty detailed work on Esopus Creek throughout the watershed, from work with Jason and Carter, showing that above and below lake suspended microplastics disappear. So the concentration in suspension in the water was dropping as you go through the lake system. So the question is, where are those suspended plastics going? And in a higher velocity environment, a creek, things that would deposit in a lake could be in suspension in that creek. So we could be sampling these same plastics, and now we see that, it looks like they are accumulating in the lake bottom, at least some of the denser ones, and that these sediments are, and we're finding that there's a place where they accumulate the most. But I think that's a good point in terms of that's kind of what led us to then think about, okay, now how are they actually concentrating in the lake system when they move out of those higher energy streams?
Another point when we were doing this, we were looking at the idea of a citizen science activity, and we had done the WAVE project with the DEC, the eel counting projects with the DEC. So this fell into that same kind of idea. We wanted to see if we can then model this event, and we needed to know where, if we collect something, what lab was it going to go to? And this is where meeting up with Ian all proved very fortunate for us. But another thing happened, and that had to do with the contamination of the drinking water for the city of Newburgh at the Stewart Air National Guard, because then we became a little concerned about drinking water sources and supplies, what's going on at Orange Lake, what's going on at Washington Lake, and what's going on at Chadwick Lake. So then Chadwick Lake became a bit of a focus, being a supply for drinking. So, all of those things played into where we are now.
Yeah, and I think I appreciate the audience being here too, because I think we're really seeing an example with Gabi's work of like, this is in some ways some very basic scientific understanding that we're advancing. Like this process of accumulation and sediment of plastics could be happening across the globe. And that's kind of how we're pitching the study. That's why we presented it before here to an international audience. But now presenting it to you as, and members of the public here in the Hudson Valley, we're saying, okay, that's great that you found this, but now tell me how do we clean it up, or what does it mean for my drinking water? And those are a lot more additional questions that we can investigate, but also as a research group, our goals are to try to find questions that can help people in an applied way, but also can help us better understand how the science is working. And I'd say the findings overall, though they're specific to the Hudson Valley, I think we're really framing them in terms of just better understanding how does the science happening around microplastics in the environment. So, yeah. So,
Emily, I can see here we have one response answering the question about the intake at Chadwick Lake, and it's mentioned here that the intake of drinking water is over a mile from the sample location at the other end of the lake. So there's a separation of about a mile from where you were sampling to where the water is taken in for the drinking.
So we're at time, and I wanted to thank Gabi and Zion and Jack so much for presenting and answering all these great questions. Thanks to you all for being here tonight and being part of this. I think it's really exciting that we have research going on right here in the Hudson River watershed that is globally important, that can add to this emerging body of research, but that also has these real local implications as well. So thanks again. I'm going to put a link in the chat to sign up for the Hudson River Watershed Alliance's email list. We're going to be doing regular student research series, speaker series, and hopefully in the months to come, we'll have more students like Gabi sharing their research that are about watersheds and water in the Hudson River watershed. So thanks, everyone. I hope you have a great night, and take care.
Thank you, Emily. Thank you, Gabi. Thank you, Zion.
Thank you so much, Emily, and thank you so much for all coming. It really means a lot.
Great work, Gabi.
Yes, very much so. Good luck to you all.
Take care.
Bye.